Okay, so let's talk about adaptations to training, right? So as we go through a training program, say 8 to 20 weeks, we want to see an increase in strength, presumably. So in blue, we have this increase in strength, and then the question becomes, what adaptations resulted in that increase in strength? And there are two main adaptations that can occur. One are adaptations to the neural system, and then followed by adaptations to the muscle proteins themselves, right? You've increased the amount of, quote, meat that you have. So which one occurs first? If you look at this graph, immediately you see some neural adaptations that kind of plateau off after about, 8 to 20 weeks. And then within, the first 6 weeks, you'll start to see an increase in this hypertrophy or an increase in the amount of protein that you have within your muscles. So the first is neural adaptations, followed by muscle protein adaptations. So when we ask the question, is hypertrophy the only adaptation that can increase muscle force? The answer is a resounding no. There are neural adaptations, and we'll discuss those specific neural adaptations in a few slides. Okay, this is a great graph to kind of explain the neural factors versus the hypertrophy, okay? So let me walk you through this graph. In number A, or in A, B, and C, the x-axis is always force production. The y-axis is the EMG. And we've seen this when we compared concentric and eccentric in the EMG lab lecture. The solid line is before training, and the dotted line or dashed line is after training. So let's look at A. And A said it's strength gain due to neural factors. So let's unpack this. Right, so first we want to see if we got a strength gain, right? Did we have an increase in force before training versus after training? And yes, we did. We see that line shift to the right. We have an increase in force production. And then the question becomes, what is that force production due to? And in this case, it was due to an increase in EMG, right? So EMG levels before training were lower than EMG levels after training. If you have an increase in EMG, you're sending more of a neural signal to recruit more motor units or increase the rate coding, right? It's a neural adaptation. It's trying to recruit whatever you have available to you. And then let's compare that to B, strength gain due to hypertrophy. And in this, you can see if you follow the two lines, there was an increase in force production, right? The line, the endpoint shifted to the right on the force line or on the horizontal line. But if you look at that EMG, the EMG did not change. So that's telling you the same neural input occurred, but those muscle fibers that it innervated must be bigger or have more sarcomeres in parallel to generate more force. So that is a gain due to hypertrophy. Typically, this will never happen alone, right? There'll always be a neural adaptation. And so C is what a strength gain would look like due to both factors. You'd have a shift, part of it just with no EMG gain, and then you'd also get an EMG gain for a large increase in force production from before to after training. So let's answer these questions. If there is an increase in force production, but no increase in EMG, what is the adaptation? And if there's an increase in force with no increase in adaptation, that has to be a structural or a hypertrophy adaptation. Number two, if there is an increase in force production with an increase in EMG, what is adaptation? And that would be a neural adaptation. So you're recruiting and increasing the rate coding of whatever motor units you have available in your body. And then number three, describe situation C, and that's the combined one. So I'll invite you to go back to the previous slide and walk yourself through the entire slide. So I've been identifying two main neuromuscular adaptations to training, but let's kind of talk a little bit more. So what are these neuromuscular factors? You have an increase in strength without changes in muscle cross-sectional area, right? So there's you're you're taking advantage to whatever muscle structure you have available to you. And so two of those neural adaptations are, as I said, increased recruitment of the motor units you have and increased rate coding of those motor units. Another neural adaptation is motor unit synchronization. So it's kind of like continuity, right? You're organizing the motor units you have to fire in the most optimal way. So they're synchronizing with each other. You also have better coordination. Again, you haven't added any hypertrophy or protein to the muscle or increased muscle structure. You've just figured out how to increase the activation of the agonist and decrease the activation of the antagonist. And then a final neural adaptation is inhibiting the Golgi tendon organs. Because if you remember, the GTOs will shut down your muscle force production because for safety reasons. So if you can inhibit or move that threshold up, you'll be able to increase force production in the muscle that you have without increasing the size or structure of that muscle. So I just want to go through some results of a study, and you're welcome to look at the original study, but I'll just highlight some of the findings. So this was a strength training article. It was in middle-aged and elderly male and females, and it was following 6 months of heavy resistance training exercise combined with explosive exercises. So you would assume they probably had a strength gain, and after 6 months, I would hypothesize that it was both neural and structural. So let's see what they found. So here are the data. They're basically the isometric force for the males and the females. Males on the top graph, females on the bottom. So this is telling you between the control period and then post 6 months of strength training, was there an increase in isometric force, right? Because if there's an increase in strength or force production, the training protocol was successful. And indeed, you can see from the control period for both ages and genders, there is an increase in force production post 6 months strength training. So now the question becomes, what is responsible for those strength gains? And in this image, we see muscle cross-sectional area. For males 40, males 70, women 40, women 70. Before and after training. And you can see there was a minimal increase in cross-sectional area. So there was some hypertrophy, some increase in cross-sectional area, but it wasn't huge. So to see if there was a neural increase or a neural adaptation, they measured EMG. So EMG for isometric leg extension. Between the control period and post-strength training, you see an increase in EMG. If you have an increase in EMG, you can assume that there was an increase in rate coding and an increase in recruitment of motor units that these individuals had available to them. So the result of this study that you've had neural adaptations and structural adaptations post 6 month training in elderly males and females. Okay, so you can summarize that the cross-sectional area of the trained muscles were minor compared to the increases in maximum strength. So strength training can be attributed to increased motor unit activation of the trained agonist muscles, increase in number of active motor units, and/or increasing in their firing frequency or their rate coding. And also decreases in the co-activation of antagonists could have occurred as well. So these are major neuromuscular or neural adaptations with this population post-strength training. So let's look at the muscular factors or muscular adaptations to training. There is hypertrophy, so you have an increase in cross-sectional area of muscle fibers. Hypertrophy accounts for about 50% of strength differences between individuals, which sounds like a lot, but if you think about it, it also doesn't account for 50% of strength differences. So what else accounts for it? Neural differences, architectural differences, length differences. So there's a lot of differences beside hypertrophy that contribute to strength between individuals. Moment arm could be another one. There's also hyperplasia, as I mentioned before, this increase in the number of muscle fibers. I had a grad student a few years back look at hyperplasia, and there seems to be evidence that hyperplasia can occur following weight training. I just don't know the recent literature on there, but it is an option. Another adaptation, which is very important, are length changes, right? We all like to stretch, except me. And so what is happening? If we're stretching, we should be assuming that you're increasing the number of sarcomeres and series, increasing the velocity potential of that muscle, which will increase the power production in that muscle, right? And so here's some basic science about chronic stretch. And what they did is they took rats and they took their gastrocsoleus muscle and they either lengthened it, so basically put them into dorsiflexion and casted them there, or they put them into plantar flexion and shortened the muscle and casted them there. And these are the data that they got following that chronic stretch period. All right, so in the lengthened condition, there was an increase in sarcomere number by 20%. So chronic stretch of a muscle does increase sarcomere number. All right, in the shortened position, you decrease the sarcomere number by 40%. So now you look at the lengthened versus the shortened and say, which is the bigger stimulus? And the bigger stimulus is the shortened posture. So to take that out to the human population, you should be worried when your clients have chronic postures that put muscles into a shortened position and sit there for long periods of the day, because that will be a very significant adaptation that may not be able to be countered by a stretching program that they might do 3 times a week. All right, so what we do during our daily lives is probably more of a stimulus than what we do during our flexibility training. Another thing to note is that sarcomeres are typically added to the musculus tendinous junction, and that our anti-gravity muscles are the most adaptable: gastrocsoleus, quads, glutes, and hamstrings. These basic studies also showed age effects due to these lengthening and shortening protocols. In young animals, and this can translate into young people starting, strength or stretching programs very young, say gymnasts or dancers, they had when they were in a lengthened posture, they actually shortened their muscle fibers, so less sarcomeres and series, and increased the length of their tendon, right? So they still got the increased range of motion, but it was not due to a muscle fiber length, it was due to a tendon length, which could have some problems later on when they stop their sport. In older animals or older populations, when they put them in this chronic lengthening, there was an increase in muscle fiber length only. So that's a good thing about aging. If you put your older client through some sort of change in muscle fiber length, postural changes, very consistent length changes during training or during flexibility exercises, they can still get an increase in the number of sarcomeres in series or their muscle fiber length. So I briefly mentioned that we have some postural positions that we could be in all day that could be a huge chronic stimulus to populations. And one key are the wearing of high heels, right? If people wear high heels, they typically wear them all day. They are in a plantar flex position, just like those casted rats. They are shortening their gastrocnemius muscle. They will adaptively shorten that gastroc muscle, right? They will lose sarcomeres and series, which will then reduce their dorsiflexion range of motion, right? And so when you have that short gastroc and then decide to get out of your high heels and go through a dorsiflexion range of motion, you will have an increased passive tension, potentially a lengthened tib anterior, and the potential negative outcomes could be lateral ankle sprains from the increased plantar flexion or an Achilles tendon rupture. And just looking at one of our postures, a sway back posture, which is postural hip extension, posterior pelvic tilt, flexion of the lumbar spine, which will result in shortened hamstrings, decrease in the number of sarcomeres and series in our hamstrings, potentially lengthened hip flexors, lengthened lower abdominals, specifically your external oblique muscles. It could lead to some quadricep muscle atrophy, Again, you're in lumbar spine flexion. That could put your spine in a very vulnerable position. So outcomes, disc herniation due to that increase in flexion, and potential hamstring tears. So here's a question very similar to the paper that we reviewed the data on, but this is after a training program of a knee extension. So in this training program, they didn't take control and train people. What they did is they trained one leg of a person and didn't train the other leg, right? So it was an internal control. So following this training protocol, the 1 rep max increased by 14% in the trained leg. The 1 rep max increased by 7% in the untrained leg. That's pretty good, right? Don't train and have an increase in your strength. The trained leg had a 5% increase in muscle cross-sectional area, and there was no change in cross-sectional area in the untrained leg. So what are the potential adaptations? First, you had an increase in cross-sectional area in the trained leg, so that's definitely saying there's probably some hypertrophy, maybe some hyperplasia, but you're getting a structural change. However, that 5% is not explaining all of the increase in the 1 rep max of the trained leg. So that may suggest some neural adaptations. What piece of these data are also suggesting neural adaptations is that in the untrained leg, you had no cross-sectional area change, but you did have an increase by 7% of their 1 rep max. So what are those neural adaptations? Increase in motor unit recruitment, increase in motor unit rate coding, synchronization, inhibiting maybe the antagonist muscles, and altering the GTO stimulation threshold.